John will be doing the open remarks.
Yeah.
Mhm.
Everybody, let's see. Whoa. We ready? There's just more people. Okay.
Slowly they will come in. I think we could start.
Okay. Welcome, everybody, to the 2024 Life Colloquium. I said that right, by the New Jersey Academy of Science. We're really looking forward to the next couple of weeks. We're kicking off tonight, and we'll also be next week on Friday as well. We're going to hear a lot of different perspectives, research, and share a lot of great information, great science, and great perspectives over the next couple sessions from students throughout the area. It's great to see the lineup and the diversity of the students in terms of the state of New Jersey, as we are from the New Jersey Academy of Science. And it's great to see the representation throughout the state. Tonight, we're going to have a couple sessions. We're going to have a keynote speaker, Miss Tanya Sulikowski. Our moderator is going to be Dr. Kiruthika Balasubramanian, who's going to lead us through these sessions, and they're going to let me talk a little bit too tonight. So I'm going to want to share a little bit of my research in terms of diamondback terrapins and what we do. I'm looking forward to hear from Miss Mahika Jawa, Miss Divya Krishna, Mr. Kezia Ganawan, and possibly, if time's permission, we'll hear from Miss Isha Vanamala. So we're really looking forward to kicking this off. And once again, welcome to the New Jersey Academy of Sciences 2024 Life Colloquium. All right, here we go.
Okay. Good evening, one and all present here.
Yeah, I might have to share here.
Mhm.
Let's see if I could, whoop, I might have to figure out a way. Whoop, here comes somebody else too.
Shall I start speaking, John?
Oh, yep, can we, whoop,
What?
I don't think I
What
could
happened?
share.
You're not able to share?
No.
Okay. So
Okay.
I'll go with the introductions.
Do you want to
Mhm.
share something?
Yeah, I'm going to share the presentation.
Okay.
Oh, I'll just give an introduction. So good evening, one and all present here. I'm Kiruthika Balasubramanian. I'm a researcher from MSK and Secretary of New Jersey Academy of Science. This evening, we'll be highlighting two individuals in the forefront of innovative STEM education in New Jersey. And we will also be featuring few presentations from students. So I do recognize in our Zoom room some of the parents or guardians of our speakers for next week who might not be familiar with the New Jersey Academy of Science. So to begin, the New Jersey Academy of Science is a non-profit organization whose mission is to preserve and promote the advancement of STEM-related fields among the scientific community. The New Jersey Academy of Science was founded in 1954, is affiliated with the American Association for the Advancement of Science, and is a member of the National Association of Academy of Science. So our New Jersey Academy of Science Life Colloquium is an initiative that aims at empowering middle school and high school, even elementary school, by recognizing their perspectives and accomplishments using diverse scientific formats. So we will also acknowledge the work of the two adult role models who facilitate these type of accomplishments. Our first presenter is John, our John, because he is the treasurer of New Jersey Academy of Science. To introduce John, John Wnek is a supervisor of science and research at the Marine Academy of Technology and Environmental Science, New Jersey, and an adjunct faculty member at both Keene and Stockton universities. John serves as a co-chair of the Save Barnegat Bay Student Grant Program and is a student research project coordinator. He is also a co-principal investigator of Barnegat Bay Diamond Black Terrapin Research Project at Barnegat Bay. where he studies the nesting ecology of diamond black terrapin. John is a co-chair of the Diamond Black Terrapin Working Group and national initiative to promote terrapin conservation, research, and education. John has several publications centered around terrapin nesting ecology, local terrapin populations, and coastal dune ecosystems. He has written several lessons incorporating the Barnegat Bay watershed as theme, and he has written curriculum that focuses on Barnegat Bay in oceanography, aquatic ecology, and environmental science. He is also working on projects to remove derelict fishing gear from Barnegat Bay waters and determining the effectiveness of bycatch reduction devices on blue crab traps. So let's welcome John
Well,
for
thank
his
you.
first presentation.
Thank you. Can everybody hear?
Yep.
Yeah.
All right. So thank you for letting me share a little bit about what we do here in terms of terrapins. I'm going to kind of put in a couple aspects of conservation along with some of the research that we do. But first, an overview of the turtle that I work with. Diamondback terrapin is a species that lives in estuaries, that mixture of salt and fresh water. They basically range from Wellfleet, Massachusetts, up on the Cape, all the way around the peninsula of Florida into parts of Texas. We think they probably go into northern Mexico, but nobody's actually studied them. They live, once again, exclusively in those type of habitats that feature salt marshes. Down in southern Florida, mangroves, there's a subspecies that's very specific to that type of habitat. But we have them right here along coastal New Jersey, and also up in some areas of northern New Jersey. So if you're up in the Meadowlands area, there's a population up there as well. Once again, salt marsh habitat is the key for these terrapin species to survive. They live as hatchlings and yearlings in those marsh grasses, and they move to open water, especially if they're females, as they get older. Males will kind of stay around the same habitat. So if they're basically yearlings, which means that they get beyond that initial hatchling stage and they grow, but they're not really kind of venturing out too much. The males don't venture out as much their whole entire life. They kind of stay around the same salt marsh habitat, whereas females will try to find nesting sites or nesting habitats overall. And this is what a terrapin habitat may look like if you see them. The unique turtle crossing sign in the middle of a marsh down here in southern New Jersey, and also the embankments that we see along the bay. That picture on the bottom right is basically a picture along Barnegat Bay, especially after the winter months and we get those severe storms and ice at times. I know we get it occasionally. We used to get it more frequently, but that helps to kind of break down the marsh, which is not a really great thing for the terrapins. But we're going to talk about that a little bit as well. And you can see where I'm talking about where it's kind of broken down. One of the things that we're keeping an eye on is sea level rise. The estuaries along the coastal areas are experiencing changes as a result of climate change. And one such is sea level rise that's creating higher water levels. And that may impact terrapin habitat. It has in some cases. And we are studying what some of these impacts are. And I'll probably hit upon a few of those as we kind of go along. Let's make sure I get that. Okay, so a little bit more about the terrapins and what they eat. They basically live, like I said, in that marsh environment. The life expectancy can vary. It's lower in the southern states where they're more active more often, and it's higher in the northern states or the higher latitudes where they're less active when it's colder, more active when it's warmer. So basically a terrapin up in the Massachusetts area, northern part of the range, will live longer than a terrapin that would live in the mid-Atlantic range. And the same is true for a terrapin down in Florida. They live a lot shorter of a lifespan. In our area, it's really hard to say. I mean, people ask me all the time. I work with, other researchers in the field, that some of them working on terrapins longer than I have. And it's basically about we say about 40 years or so. They do feed on species that include snails, fish, mussels, crabs, anything you can kind of catch out in the bay systems. So what we do is, besides testing, the environment in which they work in terms of like finding out what they're eating, by watching them eat, or when they come up and they nest sometimes, or we capture them out in the field and assessing directly what they eat. That could be a little more invasive. You kind of have to do something called the stomach flush, which is once again, not something desirable to do. It takes a lot of effort, and it's also stressful to the animals, and that's one thing we don't want to be. So what we're using is a tool called stable isotopic analysis. What we basically do is we basically look at isotopic forms of nitrogen and carbon. So those of you, chemistry, learned about isotopes, and there is a ratio of carbon to nitrogen that are found in possible food items. We call them prey items in terrapins. And then we could take tissue samples or blood samples from the terrapins that we capture, and we could kind of analyze what those isotopic signals are. And we could also look at isotopic signals from potential prey items. And if we could match them up, we could basically get a good fix on their diet. I'll go back again. So some of those species that I had there before, we're getting signals from. And on the left-hand side, what those areas that are kind of highlighted or circled shows a terrapin that's a female mature on the top. And you can see these other hoops. There's one that shows the males. And then there's another one that's somewhat green and elliptical that kind of overlaps with the males, and they're immature females. So these are what they call feeding niches, or the type of prey that they're eating. So what we're seeing is a difference in what the females that are mature are eating versus the males and immature females. And that's a way we could determine that overall, besides, actually doing some of the other analysis that we do. Males and females are very different in terms of the way they appear when they're mature. Males are smaller than females. They also have a longer, thicker tail. We call that the vent or cloacal opening, but it's a longer, thicker tail. The one on the left that we have the arrow pointed to the male, and then the one on the right, you can see she's a little bit bigger, but she has kind of a shorter, kind of stubbier tail for her size. In our area, in New Jersey, they could reach maturity at 3 to 7 years. We found some a little bit sooner if you head start them, raise them in captivity. And females, we've seen some even as young as 6 years that were mature, and as old as 11 years old. So the question here, if somebody wants to put it in the chat, is which one in that picture is the male and which one would be the female? So someone will put that in the chat. You could do that as well. Kind of a famous picture out in the field. So the top one would be the male. So the life cycle of the adult is basically, if it's a female, it's going to come up in the summer months, really late spring into the summer. Dig nests, lay eggs. The eggs will incubate. In the fall, the adults are going to, they're going to feed all throughout the summer into the fall, and then the adults will burrow in the mud. And then spring occurs. They'll come out of the mud like around this time of year, and it was warm the past couple of days. So we're actually getting reports of some terrapin activity as well. And then they'll go through their whole cycle again in terms of nesting. In terms of the hatchlings, they'll be eggs and embryos in the nest that develop throughout the summer months. Hatchlings will emerge in the late summer, early fall, and then they'll get established in the system, kind of find a place to overwinter. Sometimes they'll just stay in the same nest. There's no advantage of going out beyond the nest. And then they'll emerge in the spring, usually on days like this. Like I said, we've been getting calls, and they'll go into the marshes and into the water and so forth. So as far as that goes, we in our area have found I put a question mark by it, to 22 eggs per clutch. A clutch is a group of eggs, or a amount of eggs that a female deposits in a single kind of nesting session. The reason why I put a question mark by 2 is that sometimes we get females that get a little stressed during nesting. They don't finish nesting. And what they'll do is they'll go back to the water, and then they'll come back again and try to attempt it at a different time. And we think that that could be a female that maybe dug a nest, laid the eggs, and it was like an incomplete nesting. And she came back and had to finish, and that's why we see such a low amount, because it doesn't make sense energetics-wise why a species like that would, just deposit like 2 eggs. But in some locations, we could get up to 22 eggs. Where we work along central New Jersey, our highest clutch output was 20 eggs that we've seen and documented over multiple times. And sometimes the terrapin can come back 3 times in the same season, basically on 2-week intervals, to deposit a clutch. And that's not very common. And once again, it's a very low percentage of females that nest. Most nest 1 time, and then less nest the second time, and so forth. And a little bit more, here's a nice little cutout picture we ended up getting of a nest cavity where the hatchlings are kind of emerging on their way up from the nest cavity. So we wanted to show you what a nest cavity looked like. They're basically down about, I'm going to say more like 6 inches to, less than a foot in our area, where they nest. I rarely find any that were, near a foot down. But once again, if the sand is soft, then it's easy to dig. The females that dig the nest can dig deeper. And to complete this whole process takes about 30 minutes for a female to come up, choose a site, dig, nest, and then she completely covers the site back the way she found it. Incubation temperature is very important. It could take up to 90 days or more for the eggs to incubate. In general, it's about 2 months. If it's what we call a regular, normal summer with normal precipitation and normal temperature patterns. If it's a warmer summer, it might be shorter than 60 days, but not by much, but it would be. And if you got a cooler, wetter summer, it's going to take more like the 90+ days. But they have something called temperature-dependent sex determination or environmental sex determination, where the temperature will determine if they are males or females. So we conducted a number of studies on that. This was determined by a number of other researchers, going back a few decades ago with terrapins, and as far as what that pivotal temperature is, which is around 29 degrees Celsius. So if you have a nest that's 29 degrees Celsius, you're going to get a combination of both males and females, and that's somewhere around like 85, 86 Fahrenheit. And here's something we pulled up from a study we did a little while back, but it shows the temperature that would produce females and the temperature that would produce males. And we took this from a nest on one of our nesting areas, which is called North Sedge Island, and documented these temperatures. Looks like a lot of lines, but I have the arrows kind of pointing to the lines that are the ones that we're looking at. So basically within the nest, this looked at or measured temperatures at the top of the nest where the eggs were versus the bottom of the nest where the bottom eggs were. And the bottom eggs did not reach that pivotal temperature, whereas the top eggs closer to the surface reached that pivotal temperature. So they were more likely to be females in the top part of the nest and males in the bottom part of the nest. So even within a nest, you can get different genders, but that's not very common. Once the hatchlings emerge, they kind of break out. We got some of these close-ups where you can see hatchlings coming out of their eggs. Their eggs are cleidoic, which means that they're kind of spongy. And as the hatchlings are developing, or at that time they're called embryos, as they're developing, they're going to expand in the egg. They're also going to conduct gas exchange. So they're going to be taking oxygen down into the nest and giving off CO2. And they're shallow enough where, that could easily diffuse through the sand or the soil and, right up into the atmosphere. And they're able to kind of break out of these eggs. These eggs become papery, and they use that little egg tooth that I have circled there on the bottom left picture to kind of break out of the eggs. And they'll have what they call this reserve that they live off of. And you can see it circled there on the right, and that'll give them enough for at least 2 weeks, if not more, without having to really feed. They'll get themselves established in those marsh habitats that we were talking about before. And here's a picture of one that's emerging. And terrapins will emerge, but they'll emerge in, I would say, not consistently, as far as like, I work with sea turtles, and sea turtles, we would get a majority of them all come up at the same time. And sometimes the sand would cave in and move in it. They call it a boil. Terrapins, they may come up, one, today, a couple the next day. It just depends. So they're not they don't come up uniformly, which makes it a little more challenging when you're studying in the field. One of the questions we always ask: where are those juveniles going, or where are those hatchlings going and becoming juveniles, and how long do they spend in the salt marsh? And that's still unknown. With better monitoring, tracking devices, we'll be able to figure that out. But we know where they go within the marsh, but we're not very sure about how long they need to stay, and what habitats within that marsh itself, that we're going to find some of these terrapins, throughout the different seasons. We come across them in sometimes material that kind of washes up on the marsh, and they'll kind of get buried in that, and they'll live in that for a while. Sometimes they'll go up off the marsh into where the land is higher and it doesn't get submerged as much, or the chance of it is very low to get submerged, and they'll dig themselves in the soils there. So we call this the missing years, and that's something I'm not working on, but maybe students down the line will work on that too. And I have a number of students kind of working on different things, but not this. As far as their legal status with terrapins, you can see it ranges. The more north you go, the more threatened or endangered they are, because their habitat's becoming less and less available, and they're limited in terms of the habitat in those areas. You go beyond Massachusetts, north of that, it's more rocky type coastal areas, and that's just not the habitat that they can survive in for a long period of time. And they have all these different designations. New Jersey, I have special concern with a question mark, because they are a candidate for special concern, but it has to be signed off yet to be official. So they're a species of interest. They're designated to be special concern. That's going to happen, whenever they sign off on that. Lots of natural threats. You could see anything from raccoon that go after juveniles, maybe, some males, depending on how large the raccoon is. Definitely eggs, definitely hatchlings. As a matter of fact, in the middle, those eggs were predated. It looks like for us, looking at this, I would say a crow, because a crow will actually wait for a female to nest, and then the crow will go in, take eggs from the nest, and then, one by one, fly away with them and then devour them, or kind of stay close to the nest site and also, kind of retrieve whatever they can from that. Eagle, we've seen some eagle. In our area in central New Jersey, we're seeing an increased population. By eagle their nests, we've seen remains of terrapins. Blue crabs go after young ones, and fox. We have a red fox population, and even coyotes in our area, because I live adjacent to Long Beach Island, and there's reports of them as well. And they will also go after some of the nests, and even some of the adults, and juveniles. These human and terrapins living together, these anthropogenic threats. So that's one of the things that we're working on, is trying to, have us coexist. You can see this area here on the right-hand side, that is a lagoon. That's like an area that's kind of walled off. You can see the boats that are all in that little channel, homes right on that. And if you look on the left, it was more than likely that area on the right kind of looked like that area on the left, which is a salt marsh habitat. But these salt marsh habitats are also eroding. All of this erosion, all of these, buildings, building of these walled areas or channels, we call them bulkheads in our area, have cut off like a lot of the nesting areas. And there's a lot more erosion taking place and loss of these areas. So we're kind of working on that. There's other threats. We have these devices called crab pots that you throw in the water. You can leave them in for 24 hours or even longer. You'll put bait in there. They'll catch crabs. Sadly enough, the terrapins like the bait. They like the crabs that are in there. That's, one of their prey items is a blue crab, the crabs that we like to catch and eat. And sadly, the terrapins could get caught in those traps. And since they're turtles and reptiles that, breathe air, they need to come to the surface, and they're not able to do that. And they sometimes can drown, much like that gruesome picture on the top right. That was down in Maryland back in the early mid-2000s. And that led to the state of Maryland doing a lot more to protect terrapins. At the time, they had a harvest. And we also had a harvest in New Jersey up until 2016. So, this kind of stopped the harvest in Maryland, because of the public outcry, this net with all those terrapins. And then the other kind of gruesome picture on the bottom right is a terrapin that, sadly enough, was a female coming up to make a nest. In the areas that they nest, sometimes they come back to those same areas that they come back every year. Usually, it could even be an area that they were hatchlings that emerged from, and then they come back to that same area. There's roadways in the way, and it's not a good, that's not a good scene. That's something we're working on as well. Also, boat propeller injuries. That's another type of issue terrapins have to deal with. These are just some study sites. I'm not going to keep it up there long. This is Barnegat Bay. There's New Jersey, and you could see Barnegat Bay itself with a barrier island. And these are areas that we've worked on over the years. Some of our partners, some ongoing projects. So we're starting to study the population. We're probably over 7,000 terrapins that we mark or know about in those areas. And this is what we do. We process a terrapin, we call it. We will capture them, whether they're nesting. Sometimes we'll go into areas and we'll try to mark or assess that population for size, for health. This is how we do it. We use a caliper to measure them. We kind of work with them overall. We even take blood samples. We use that for genetic analysis and for health analysis. This is Courtney, one of my field assistants. She ended up working on a thesis over at St. Joseph's University with terrapins. She was a really good one, doing a lot of this work. You could see she could basically capture a lot of terrapins that kind of made her day. She was really great in the field doing that. We assess them. We also make sure that we give them a unique code so that we know when we recapture them, that they're going to be the same turtle or terrapin. What we do is we'll use a passive integrated tag, PIT tag we call it. So it's passive integrated tag, which has a unique code, and we have a scanner. This tag is put into the rear leg area. So it's not going to be as invasive. It's small. You can see that too. It's extremely small. It's only, maybe about 10 millimeters long and maybe about a millimeter and a half wide, with a special injector that we use and an iron we can kind of read. We also, I circled that one because that was a unique turtle. Well, that's another talk some other time, but that was important. To notch or not to notch, we also notch their external shells, even though we mark them with a PIT tag. And the reason why we do that is to prevent poachers, which is another threat to them, where they're kind of taken and sold in the illegal pet trade. We notch their external shells. You could see the codes that we have around there. Each one of these, we call them scoots. They're little plates that go around the outside, and you can see the lettering system we have from A to X. And what we could do is give them a unique code and notch them. So now they'll have a notch and a PIT tag, which makes it a little bit more unique and identifiable, specific to the area, location, and time. And we could monitor growth. We can monitor recapture and so forth. So here's some of the areas we're doing some work. We have about 1,600 terrapins marked local to where I am right now in one of the study sites. We're going to be actually up with that. Oh, I left this one in here. This one's kind of cool. This is a male, and we give them those unique codes. We're up to actually 7 letters now. And they also get a PIT tag, and we used to get pictures like this of them, but it became a little bit more difficult to maintain all those. But we still do get blood samples when we can. Some of the terrapins we outfit with loggers. This is a what we call archival tag. This does depth and temperature. We're able to fit this, and one of the hopes we have is that once we mark this terrapin or we put this tag on this terrapin, this will collect temperature and depth throughout the entire year. And if we recapture it again, we could actually extract that data and get data like this. Basically, what this graph is showing is something called brumation. It's a form of hibernation that reptiles undergo. So some mammals undergo hibernation where they shut their whole body down in terms of metabolism, and they consider it like a sleep. Terrapins will reduce their body activity, but when it gets warmer, they could actually come out and be active. And then when it gets colder, they can go back into that inactive phase again. This kind of just gives us the temperatures at which during brumation, these terrapins are exposed to or they're undergoing when they're down in the muds. And you can see at times it approached zero during the true brumation. You can see that lower area boxed out. And also we were able to look at depth. How deep were they at times? And its pattern is kind of up and down like that because they are in areas along the coast. So the tides influence the water level above them, and storms can have more water. You could have extreme winds that are blowing water out of the bay, and we call that blowout, that could actually make them more exposed. So we're really interested in this. So this is the first terrapin that has this archival information that comes back from the year. I'm thinking of other studies in other places, but I think this is like the first one that we're able to get a whole year profile on it. And its code was BHIO, one that we marked back in 2004, and we basically see it every year. It's amazing. We're trying to help with nesting habitats as I kind of close out a little bit. So we have something called the sand game, where we're kind of pushing sand around. That's a primary type of nesting substrate or soil is sand. I mean, they're in the coastal areas. Of course, that's where you want to nest. And we're working on projects where we're kind of protecting shoreline, trying to build it back up because it's been eroded. This is a project, this was, I believe, along the Delaware, where we're seeing like a improvement, and we're seeing like a invigoration of plants and rib mussels, which are a type of bivalve, related to a little bit. They're not as related to blue mussels like the edible mussels. These are more of a marsh mussel that can kind of live in those conditions and absorb a lot of energy. So we call them living shorelines. We're putting in turtle gardens. We have to deal with this because there's a lot of development in areas where these terrapins nest. This is those blue markers over there show where the terrapins are, come to nest. Those areas that you could see from this aerial are homes, are properties. So we're trying to work with residents in these properties and townships to create nesting habitats so they're not crossing roadways or going onto people's properties and so forth. So there's a lot of projects. We're working on a project close to here. That's the one where we have 1,600 terrapins marked. You can see that area during a big storm where it flooded out. And the only area exposed over there was that where I have circled in there. So we kind of looked at this area. We had a bunch of volunteers come out. This was a Girl Scout troop that helped us to move some sand, rake some sand, put up some fencing to keep, pets out. I like dogs and cats, but, in nesting areas, they could kind of dig it up. So we want to, make sure we protect them and make sure people aren't walking across and so forth. So we did that and kind of made it look like this. It becomes a little bit more of a nesting spot with some vegetation. Same thing over here in one area where these terrapins were getting hit by cars. We said, hey, why don't we just put a nesting area to keep them away from the parking lot, because they were going in the parking lot and trying to nest there, and the cars, were accidentally kind of running over them. And basically a pile of sand, and, with some of our Project Terrapin volunteers, we can do this. Major project over in Little Lake Harbor, an area that has probably the most dense nesting area that I've seen in New Jersey, maybe down in Smithville, Brigantine area, which is south of here, close to Atlantic City, may have a more dense nesting population. But this place, if you go here during nesting season, you would really have a difficult time trying to get step over terrapins because they just come up in such high densities. But anyway, they took this area that was circled at the top left, added some sand in the bottom left, and that's Ben Wurst with Conserve Wildlife, one of our partners, made this massive nesting area with 90 truckloads or dump truck loads of sand. So we do this. We protect some of the nests. We assess some of the nests. You can see there's a nest cavity in the bottom left that has some eggs. And that's Kira that was working with us. She was a volunteer for a number of summers. monitoring the nest. You see how the eggs set up in there. We put protective covers over them to keep out the large mammals, especially the fox and the raccoon. We also have smaller nest covers that the residents kind of like to use on their properties. If they have a terrapin up nesting, we give them a chance. Big open mesh so that the hatchlings could kind of get out on their own and we don't have to keep them trapped in there, which is, not ideal and not at all something that we would even consider. But education, like we're doing today, talking to all of you is a key. We're working on it. We have a program going. We're proud of it. It's been going for the past 16 years now. Wow, I can't believe that. And we distributed over 30,000 bycatch reduction devices, these little inserts that go in crab pots that actually help to reduce the probability that terrapins are going to get in there. Crabs still could get in, but the terrapins are less likely to get in. So that's something we're working with the state of New Jersey on. And road signs, we have a road sign contest out now where we're developing a new kind of lawn sign. We do that every so often. Hatchling releases. A couple of our students, or a few of our students from the past, there's we're part of a program where we're recovering some of these abandoned crab pots that could actually still capture, not only crabs and fish, but also terrapins, and they're considered ghost pots. We have the BRD program, as you see there. Those are those bycatch reduction devices, those orange rectangles on the left that go in the crab pot. Like I said, we're giving those away. We get them funded, and it's part of what we do. We do that because if you put a BRD in, this is kind of showing all those blue little squares are showing terrapins that we found in a trap that were dead. And we said if we put the BRDs, had the BRDs been in the trap, those that were circled there would have still not been able to get in the trap. And, they probably would have lived, of course, who knows what would have happened to them after, but that would have kept those out. So that's something we're working on. And that's how that works. I have that picture again there showing that. And what you can do, and that's how I wrap up. Once again, we have something called the Turtle Gardens Project. We're working not only locally, which I'm really so excited about because some residents are doing this. I don't have a picture in here of a resident, but they took half of their property and they put this turtle garden in. And it's amazing. They maintain 40 to 50 nests a year, and it keeps them off the roads and out of the neighbors' houses. And it's a great marsh they can go to. We're working on Long Beach Island with these turtle gardens. We're working with people out of state. We're working with the Inland Bays program in Delaware. And now we're going to work with the Maryland Inland Bays program. We're going to be meeting with them on Monday to just talk about a lot of this stuff. The key thing I always tell you, notice the wildlife around you. No matter where you live in New Jersey, you still will see wildlife. And just notice it and be aware of it. And I think awareness is like the first step overall. And support all those efforts in going out and doing cleanups. In our area, we do beach grass planting. That all helps to preserve our dune systems, our barrier islands. And that's so important because it protects that terrapin nesting habitat. And it also protects those salt marshes where they live and some of the volunteer monitoring. I put this up here because these are some of our students from Mates that were part of our Head Start program where we raised them in schools, raised them in our classroom. They were the first group that went through that. They're all out in the field doing great things now. One of them, I know she got her master's working with terrapins. So that was a little while ago, but I put that in because that, kind of shows, those efforts and what you can do to help. So it doesn't matter what age you are, you can make a difference no matter what age you are. Really kind of stepping up, as a lot of you know. And these are our partners, and we always thank them. And once again, representing Project Terrapin and the Diamondback Terrapin Working Group, thank you for listening to all of this. Please help us coexist. And, I think we'll do a question session when it happens. I just like this picture. Thank you.
Thank you so much, John. Do we have any questions for John? Students, if you have your questions, you can just unmute and ask directly.
We got Jason. Go ahead, Jason.
Yeah, I know this is pretty obvious, but I found it, keep in mind, I am not really an expert in the area, but I found it interesting that they don't seem to have a social family structure. Like, normally you'd expect like the mother to take care of the children and to feed them for a while and to introduce them to the intricacies of life. So they just develop independently, huh?
Yeah, that's true too. The only parental investment is the mother coming up, digging the nest, covering the nest, and then she goes back. What I find interesting though is for years doing this work, there were some terrapins that when the female came up to nest, it was always at the same time, exact time, with another female that came up to nest. And each year that happened over multiple years, the same pair came back. So I think there's some kind of dynamic with that later on where they kind of bond in some way. I don't want to like overstretch a little bit, but I see that in here. And it's a little different because we have a number in captivity that can't be released back to the wild. They wouldn't be able to survive. So we provide care for them here. And they have a type of bond where, you know, we had one that just wouldn't feed unless the other one was there. So as far as parental investment, none, but beyond that, that kind of behavior, I really think could be studied. I really think there's something there.
Any other questions? Yeah,
John,
Scott. Yes.
go ahead. I have a
Hi.
question.
Oh, who's up?
Go ahead.
Hi. First of all, very interesting talk. Thank you very much. I have a quick question about the temperature sex determination, and I apologize if you talked about this. I had a dog at the doorbell kind of thing happening when you were talking about that. But I was thinking about the you know, with warming temperatures and things like this. You know, obviously that's a conservation concern, right? So do you guys note whether or not the females, you know, behaviorally modulate that? So like if it's really a warmer, you know, breeding season, they bury deeper, you know, or something like that, or?
Yeah, we've studied that. In fact, we're analyzing some data from an area where we compared terrapins nesting in like residential areas on properties, basically, versus like our turtle gardens areas that are more open sand. So it's not as compacted to see if there's a difference in the nest depth. And we compared it to their leg length to see if if there's anything to that. What we found is it's easier for them to dig in the sand. So they'll tend to dig in the sand more. So in response to your question, by having these turtle gardens or installing these turtle gardens, if we're seeing some kind of shifts where like the males in some of our areas could actually be responsible for more than, for, we call it multiple paternity. So a nest could have multiple males. If we start to see that kind of shift a little bit where the genetics from one of the males is more prevalent, in an area, is becoming more focused, we will then, think about those practices where the female could dig deeper in that loose sand in those turtle gardens. And that may be an indication of, cooler temperatures that would promote more male, versus the warming that's taking place, where they can't dig as deep and they're going to be more skewed towards even more female. That it favors female development, this strategy, but it could swing too far, that way. But we monitor those nests using temperature sensors, or we call them eye buttons, and we analyze that, over time. And we have a question from Jason, I think, saying, do you provide food? Oh, absolutely. We try to provide what we can that's more, like natural food, like fish or shrimp. We definitely in a captivity give them more restaurant quality stuff, fish or shrimp, but not, we don't take it from the wild to give to them when they're in captivity because we're afraid of the diseases that they could contract from things out in the wild and them not being out there.
John, I have a question. If some students who are listening to your talk, if they are interested to join summer program with you in your project,
yeah,
how should they approach, or what are the qualifications you're looking for?
Yeah, well, for students that are in high school, I've had students come down from high school to kind of work with us. Matter of fact, we had one, recently from out Western New Jersey for a couple summers, and then, she went on to school. I think she went up to Cornell. But we do. So if you go through projectterrapin.org, and contact me, my email's there. I had it in the beginning too. I could put it in the chat. We could kind of direct you in that way. So that's probably my best answer for that. Because there's so many multiple things. I have like summer contingent of high school students, a summer contingent of college students, and then we also work with some grad students and beyond, depending on, the projects we have going on.
Hi, John. This is Maria. How's everything? Great work. Oh my God, like I love your work.
Thanks.
I had the honor to actually go to John's lab, and oh my God, amazing. So my question here for you, everything was excellent, and it was more about the advocate effort that we should be addressing for terrapins, right? I
Yes.
mentioned the fact that we have Massachusetts, Connecticut, New York having that a special concern, New Jersey maybe in a question mark, and we're just waiting for something to be signed. How can
Yeah.
we help to get
How can we help? That's a great question. One of my friends is also in charge of the endangered and non-game species program in New Jersey and he handles reptiles. He's a really good guy. And I think he's just as anxious as well. The issue, I don't want to get into a long explanation, but it was basically a majority of experts that handle reptiles and amphibians. There's a group, and it's called the Delphi process, where they evaluate turtles and also frogs, and so it's across the board. And then they come up with a recommendation, but the recommendation for terrapins was that 6 individuals felt they should be special concern or greater, and 5 felt they should be stable. So it was really close, a 6-5 vote. There were 11, I believe, in that vote. And what happened was, in the 6 that said they should be special concern or greater, some said they should be actually considered like a threatened species. So that's what they're kind of hung up on. They're not going to move them there. I'm not going to say never. That's a terrible word, but they're not going to move towards that. It's definitely special concern. But I think what they're doing is we're just waiting for like a signature. So yeah, I guess that's something we could, if it needs to be, it could be moved on. Because they're not able to be harvested in the state, it's not as much of a problem anymore. But if they were able to be harvested, I don't want to get into that. Then that designation would be very important. So right now, because they can't be harvested or possessed without a permit, it gives them that protection, and they're not moving on it, as fast.
My concern comes from like the habitat perspective, right? I seen and I have been in the the south side of New Jersey. There's so much building going on, and aside from the building going on, there is the whole plan of sand removal, which sometimes, depending on where you are at the beach, you don't know whether you are afflicting, an environment by terrapins, right? Because they are by the marshes and that areas, and they're using those areas sometimes to build.
Yes.
So, those are the way I was viewing,
Yeah, absolutely. But we're optimistic, because I'm always optimistic, because there's some, like, even in township government, county government, state government, there's some real dedicated, good people that kind of understand what's going on. And the Terrapin at least is getting some of that support. And then locally too, some of the towns, like I met with the town a couple of days ago, local here in LBI, that's going to take part of their park and make it a terrapin nesting area. And they're going to fund it and fence it the right way. And, because they know that there's less habitat for them to go in that town, and they want to make sure they do right with that. And that, that's what we're kind of very, very not only happy about, but it's motivational. It kind of shows that, these towns and the residents and stuff can make a difference. So.
Great job. Thank you.
Thank you.
John, I have one question. Like, what is the brumation? What is the period duration of that brumation process? What you said.
For the brumation, what happens is, as we get more into the fall and it gets colder, and we get less daylight, the terrapins will stop feeding. There's like a certain temperature where they feed and stop feeding. And at that point, it starts triggering something in them that they then will start to move to like a wintering area. So some will actually dig themselves into a marsh some, the actual salt marsh itself, or go under the marsh itself. There's like marsh ledges, and they'll kind of go in the underbank there. And the females will go into an area that's deep enough where the water doesn't freeze, and deep enough where when tidally the water kind of remains, they can remain submerged and not exposed, but a kind of mud that they can kind of bury themselves in. So we know females that are in those type of habitats overwintering because their shells become smooth. Sometimes we age them by their annuli, or those are like concentric rings on the top of their top shell or even their bottom shell. But over time, the sand acts like sandpaper and it smooths those over. So we even lose count of their age. But if we mark them initially, we would know their age, year after year. So that's what's great about marking them. But we see that, and that temperature and that shortened daylight time kind of triggers that. And it's usually sometime in late October, early November. And if it's a warmer fall, it could be delayed a little bit more. And then if the water warms, and they're shallow enough and it warms like this time of year, they could get a little more active. I've seen them active in April. So this would be a little bit early March, but I have seen them active in April.
Thank you so much, John.
Well, thanks for letting me share.
Any other questions? Okay. Thank you so much, John. Thank you for your presentation. We really enjoyed about other things. So now we'll be moving to our next presentation from Tanya. Into this one sec. All right. So our next speaker is Tanya Sulikowski, who is currently the Director of Education at the New Jersey School of Conservation in the heart of Stoke State Forest. Tanya trained as a field ecologist and has worked with a variety of species throughout her career. Her favorites are often less loud bats, rattlesnakes, and bugs. Benthic microinvertebrates, it is the tiny bugs that live in stream bottoms, were the focus of Tanya's master thesis research and are still a passion of hers today. She also taught middle and high school science and has used hands-on knowledge of ecology to bring science lessons to life for students. Her experiences in the forest and the classroom demonstrated what a powerful teacher nature can be. In 2017, Tanya was selected as the Grosvenor Teaching Fellow by National Geographic, and her relationship with National Geographic has resulted in several grants to help educators learn to teach about biodiversity in their classrooms. And her most recent award focuses on a new initiative called Macro Blitz. Macro Blitz will inspire and empower people to find and photograph aquatic microinvertebrates and share them through a citizen science app called iNaturalist for science and for learning. Her keynote for the New Jersey Academy of Science seminar will share more about why this is so important. So in Tanya's off time, she lives what she teaches. She is an avid native plant and organic vegetable gardener and loves to hike, paddle, and explore the planet's wildest places. Let's welcome Tanya for her next presentation. We are very happy to have you, Tanya. You can
Great.
start it.
Thank you so much. It's very hard to be the bug lady when you're following the turtle guy, especially with all those pictures of baby turtles.
Yeah.
So let me just share my screen. All right. So can everyone see and hear? Okay, great. I know I have at least one bug fan in the audience. Scott Kite is here, and he was my thesis advisor on macroinvertebrates. So at least one person will love bugs just as much as baby turtles. So tonight I am going to talk about a bug's life, lessons we can learn about our environment from some tiny creatures. I am the Director of Education at the New Jersey School of Conservation, and I am honored to be here to be one of your keynote speakers tonight. I've just gotten more familiar with the work that the Academy of Science does, and I and the School of Conservation are both very excited to be a part of that. So tonight, I'm just going to, I took a slightly different approach with my presentation than John took with his. I'm going to give you a little bit of background on my career path, because I know many of you are high school and middle school students that might be starting to think about the path that you want to take. And I took kind of a circuitous route to get to where I am today, as someone who's focused on both education and research. I'll talk a little bit about our current relationship with National Geographic, which I'm really excited about. My work at the School of Conservation. We'll do a little bit of biology of bugs, and I'm going to take you on a virtual field trip, because I know that these evening meetings sometimes can feel pretty long on Zoom. So we're gonna go out of the Zoom meeting for a bit and go do a stream and lake visit. And then I will, hey, Marius, I think you just came in. If you don't mind muting, that would be great. We are going to talk a little bit about the iNaturalist app, and then I'm giving you a homework assignment. I'm not a classroom teacher anymore, so I don't get to give a whole lot of homework, but this is a fun one that I think everybody will enjoy. So first, I am pretty happy with the career choice I made. I have an undergraduate degree in wildlife ecology and a master's degree in ecology, and I have spent the last 25 years as an ecologist doing research on a variety of different species. And I got my start, you can tell I look really young in that picture in the upper left-hand corner. That's me right after graduating college from Rutgers in 1997. And my first project was working on plants. And if you look at the rest of the slides, there's a lot of other fun wildlife in there. But you know, I built my foundational knowledge and my foundational work in the field focused on plants, and they don't get nearly as much attention as they deserve. Without plants, we would not have an environment, and so many of the other species that we study depend on the relationships that they have with plants. I've done some real other interesting work. I'm there with a peregrine falcon in the picture next to that one. Right beside that, I'm doing some fisheries work at Merrill Creek Reservoir. Right beside that, I also have some experience doing turtle research. That's a wood turtle I'm holding, and we're putting a radio transmitter on the turtle to track their movement. In the lower left, I've done some work with bald eagles. That's a nestling bald eagle that we took out of the nest to take some measurements and some blood samples. I've worked with black bears and bats, and that's a leatherback sea turtle in the bottom right. So when you're a wildlife biologist, one of the benefits of being in that field is that usually you hang out with other wildlife biologists. So you don't always get to just focus on your research, you get to help out with other folks' research too. And so I feel really lucky that over the course of my career, I've had the opportunity to work on many, many different species. Also, being an educator is another cool career choice. And I was a formal classroom teacher. I worked with middle school and high school students, but the majority of my career has been focused on fieldwork and environmental education. And so the joy of doing that is working with students much like yourselves and getting them out in the field for the first time. In some cases, you know, exploring with them, learning alongside them. I always joke that I've learned more working with children than I did at getting a graduate degree, because kids ask questions that are so hard that you really have to think to know the answer. So that combination of research and education has led me down the path to where I am now. And this is going to be really the focus of my talk tonight, the macroinvertebrates, but I wanted to bring it up now because I wanted to make the point to all of you that you don't always end up where you think you're going to be. So when I did my master's thesis, I had planned on doing something really exciting, like going back to Trinidad and working with leatherback sea turtles, or going to Mexico and working with monarch butterflies. But at the time, there was grant funding, and the opportunity presented itself for me to work with macroinvertebrates, these teeny tiny little bugs that live in stream bottoms and can tell us the story about the health of our landscape based on what we find and where we find them. And so I was not opposed to the idea, but it definitely wasn't a species that I thought I would end up focusing on. And then spent 2 years collecting and identifying.
Even Violet tried to play with her, and she didn't want to play, I noticed.
Hi, if someone's on their iPhone, if you wouldn't mind muting, that would be great. So I spent 2 years collecting and identifying these species and learning a whole lot and getting a much greater appreciation. So the National Geographic connections that I've made are pretty exciting. As many of you are probably familiar with the work that National Geographic does, they invest in people who focus on science and exploration and education. And the idea is that they tell stories to help protect the wonders of our world. And in my case, when I was a teacher, I applied for a National Geographic fellowship that gave me the opportunity to meet other teachers around the world that are doing innovative science and research. And they're kindergarten through 12th grade teachers. And the idea is that, we came from all these different backgrounds, but the problems of the world are so complex that they're looking for people with all different backgrounds to help figure out the solutions and how to educate folks. So through National Geographic, I got the opportunity to travel to Alaska. There's a picture of me on the lower left. I've been to the Galapagos Islands twice with National Geographic and the Teacher Fellowship, and there I am with the Galapagos tortoise. And I got to help out with some research where we were collecting tortoise scat. So that picture in the lower left-hand corner of the main picture is my foot next to a giant piece of turtle poop.
Oh,
So,
wonderful.
research is not always super glamorous, but it's always really interesting. And then now we've received several grants to focus on our teacher professional development. And what we have been focusing on recently is partnering with National Geographic to produce this new macro blitz program. So I'm part of the education team. There in the lower right, you'll see me pictured with 4 other people, and one's not in that picture, but they're National Geographic explorers. So folks that work in Mexico and Patagonia and Canada and here in the US, and they're all entomologists and they're all bug nerds like me. And they're working together to launch this new citizen science initiative that will help get people really excited about looking for bugs and sharing information with bugs at the global scale. So I'm going to talk a little bit more about how you can get involved with that in just a few minutes. All right. So I work right now as the Director of Education at the New Jersey School of Conservation, and we're in the heart of Stoke State Forest. They're in the upper left-hand corner of New Jersey. We're surrounded by about 75,000 acres of preserved open space, which is really a gift. The Delaware Water Gap National Recreation Area, Stokes, and High Point are around us. Our school is celebrating our 75th anniversary this year, which we're very excited about. We're the oldest and largest year-round environmental ed center in the country. And what that means is that students come to us on field trips, typically for 2 or 3 days at a time, and spend time outdoors learning and getting excited about nature. And so many of the people that are in the scientific community in New Jersey, or really our environmental leaders, got their foundation and had their most exciting childhood learning experiences at the School of Conservation. So we're really happy to be there doing the work that we're doing. My job is wonderful. I always say it's the best job in the world because I get to not only be out in the field with kids and focus on that outdoor learning, but I also get to work with teachers to help them learn how to take their students outside. A new development for us is that we are now the Northern New Jersey field station for Rutgers University. So another really important and exciting part of my job is that I get to work with researchers that are on site doing research and help them connect the research that they're doing with the teachers and the students that come to visit us. And so the School of Conservation is a unique place because we do all 3 of those things. We work with students, we work with teachers, and we do our own on-site research. All right. And this is the transition to the meaty part of my talk. So water ecology is our most popular field experience at the School of Conservation. And as an ecologist, I love to teach this class. We find all kinds of fun things with the students when we're out in the field. We can share this experience with learners of all ages. So you can see the littles there, kindergarten students out with nets dipping in our pond. That's a group of high school students in the middle, and they've got identification keys and microscopes and more sophisticated scientific equipment. And they're looking for things like that stonefly that's in the picture on the right. And the wonderful part about this class and the wonderful part about the homework that I'm going to give you that hopefully you'll have a chance to do is to get out and discover this world that very few people ever get to see. But it's so important to understand that it's there and to start to appreciate, as John said, and just look around and recognize the things that are sharing the world with us. And the wonderful thing about being able to do that is that these insects have a story that they can tell us, and we just have to know how to listen. So the benthic macroinvertebrates that we're talking about tonight are basically, I'll break this down for you so you can be with me on the definition. Benthic is something that inhabits the bottom of a stream or the substrate that it's living in. Freshwater is stream, lakes, rivers, and ponds. I think we all agree on that. Macroinvertebrates are relatively large, so generally you can see them with the naked eye. Many of the species that I worked with on my thesis were about the size of an eyelash, but still, if you dip a net in the water and pull them up, you can see them wiggling with your naked eye. And then an invertebrate, obviously an animal without vertebrae or a backbone. So that is the focus of my thesis research and also the focus of what we're going to talk about tonight. So just a quick review of life cycle. If you haven't thought about insect life cycles in a while, insects go through either complete or incomplete metamorphosis. The caddisfly example on the left basically shows the eggs are laid by the adult, they hatch and form larvae and then move into some sort of pupa stage that then the adults hatch out of. The difference between that and the incomplete metamorphosis, like the dragonfly example on the right, is that there are only 3 stages to that life cycle. The eggs hatch and look like a nymph, which is sort of like a smaller version of the adult. And in this case, that weird looking creature on the right-hand side is a dragonfly nymph, and it looks a lot like the adult version of the dragonfly would look without the wings. And so when we are sampling for these critters in the streams, most of the time, the things that we are finding are the larva or the nymph stage of the metamorphosis. And if anybody has any questions, feel free to just pop them in the chat. We'll have time for some questions at the end, but if anything comes up as we're talking, feel free to ask. So this is the story of how insects can tell us what's going on with our landscape. And I'm encouraging you to take a screenshot of this biotic index and these 2 questions, because in a moment, we're going to take our field trip. And I would like you to be thinking about the things that we're finding on our field trip as they relate to this biotic index. So the concept is fairly simple, but it's a really powerful tool for citizen science. So if you look at the top row of critters that we've got there, they can be classified as group 1 organisms. And the caddisfly, the mayfly, stonefly, water penny, riffle beetle, Dobson fly, they are all indicators of really good water quality. They can't tolerate any pollution at all. So if you are in a stream that has had some kind of runoff or some kind of pollutant, you're not going to find these organisms. They are absolutely required to live in really clean, pure water. If you look at the second group of organisms, the dragonflies, the damselflies, the crayfish, the scuds, the clam, the sowbug, those organisms can tolerate some pollution. They still need pretty clean water, but they're a little bit more tolerant of degraded water conditions. And then the third group, the group 3 organisms like leeches and midge flies and aquatic worms and mosquitoes and black flies, they're all very tolerant of pollution. So if you were sampling in urban areas that have a lot of roadway runoff or a lot of development, those are organisms that you might find in those kinds of waterways because they are much higher, they have a much higher level of pollution tolerance. And another thing just to sort of keep in mind is any of the group 3 or group 2 organisms can also live alongside the group 1s because they also like clean water, but they have a much higher pollution tolerance. Does this make sense to everybody? Everybody feeling good about this? Give me a thumbs up if there's any questions. I think everybody's good. All right, cool. So during our field trip, which we're going to take in just a moment, I want you to keep notes on the organisms that we find and if they are group 1, group 2, or group 3. So you should have a pen or piece of paper or we're in a tech age. You can use your cell phone to write down the organisms that we find. But again, if you haven't taken a screenshot of this picture yet, I would encourage you to do that because it will help you as we are traveling along on our trip. All right. Oh, I'm having a little slide advancing issue. Hang on one sec. There we go. Okay, so before you go out in the field, before we go on a field trip, you need to make sure that you are ready to go. And the gear that you need to do this kind of science is not incredibly expensive or complicated. And that's one of the things I love about being an ecologist. Most of the time when I'm going out to do field work, I can grab what I need from my kitchen and make do with it. And that's exactly the case for this kind of organism sampling. So you need a net or a strainer, and I've got a fish tank net here, which is an awesome tool. And I also have a kitchen strainer, which my husband always gets annoyed when things disappear from our kitchen. But if you have something that you strain spaghetti with pretty low mesh size, that's a great tool for collecting. A tub for keeping things is awesome. I've got this white one, so there's a lot of contrast. And if you put organisms in there, it's easy to see them kind of wiggling around. A spoon. Again, really sophisticated piece of scientific equipment, but this is super handy to pick things up and be able to get a good look at them. And we'll do that in just a minute. And we usually wear rubber boots when we go out in the stream. You can get a pair at Walmart for like $10, but old sneakers also work really well. There's all kinds of identification keys. I have a personal favorite, but I'm at a high nerd level on identification. There's lots of books. There's lots of one-page dichotomous keys out there online that you can look at. And there are also some really good apps, which I'll talk about in just a minute. But most of all, you just need an adventurous spirit. So part of your challenge for your homework is going to be to find a local waterway and go out and take a look for some of these things. Once you get to a slightly higher level, and you want to know more or see more details or learn more about these individual organisms, things like a ruler and a petri dish might be handy. A hand lens is a really great thing to have. Because you can zoom in and look at the organism that you're looking at. Ice cube trays are great for separating different specimens that you might be collecting. And then if you get really fancy schmancy, you can invest in a field scope, one like this that's rechargeable that you can take out in the field with you, only costs like $150. And you can take it out and have it right on the stream side while you're out there working. And then if you have a phone on or if you have a camera on your phone, that's another really great tool, especially for the Macro Blitz project that we're going to talk about in just a few minutes. So you don't need a lot of equipment, and you can go out and do some really good science without knowing a lot of anything about this before you start. Sorry, I'm having some slide issues here. Okay, so our first field trip is we are visiting a lentic habitat. And lentic means mostly still water, as opposed to lodic, which is our second field trip, where the water will be flowing. And if you have a hard time remembering the difference between lentic and lodic, I always remember lodic sounds like flowing. So these are the two different places that we're going to visit, and get ready to take some notes on what we find.
The audio volume is very less. I can't hear it.
Hi, Tanya. There's no sound on the video.
Did someone say there's no sound?
Yeah, yeah.
Let me try it without full screen. Let's see if that works.
You may have to put on the side to use the audio. Sometimes there is some technical difficulties, but it was fine when we I think when you share it, maybe that might be the issue. Let's try one more time.
Okay. Can you all hear that now?
No.
Oh,
All right, hang in there. So I tested this earlier and it worked. Does anybody have a suggestion for something I can try? All right.
You may do a share sound maybe. So we having
Sorry, everyone. Bear with me a sec. Okay, so I'm going to put the closed captions on. I think that might be the best we can do. Let's try that. Can everybody see the closed captions?
yeah. All right. So that's our first field trip.
So our second field trip is going to be visiting the Flatbrook River, and I'll turn the closed captions on for this one too. My apologies. And we'll see what specimens we can get here. Okay, everybody. So sorry for the technology challenges. Unfortunately, this is the world we're living in. All right. So you had an opportunity to look at some of the things we were catching on our virtual field trip. And what are some of the things that we found in Lake Wapelani? That was our first stop. Can anybody name any of the organisms that we were able to find while we were there? Just throw them in the chat. All right, so I'll help you out. We found a dragonfly. We found that red-spotted newt. We found a sow bug or an aquatic isopod, and we also found a damselfly. And so what group of organisms? Yes, we got a mayfly. Awesome. Thanks, you, Isha. Any ideas what group of organisms most of those fell into? Second, sweet. And yes, I have gotten bit by things out in the field. That giant hellgrammite that we found in the flowing stream can deliver a little bit of a bite, but it's not too much to worry about. And then in our flowing habitat, we found a whole variety of other organisms too. Does anyone have any notes about what we found in that flowing stream, the Lodic habitat? Awesome. So we did. Thanks, Mike. We had a stonefly. We had, and that was the one I got really excited about on the underside of the rock. We found that giant Dobson fly larva. It's also called a hellgrammite, and we found some caddisflies. And so the story that we are told by the critters in that landscape are that basically our water quality for the stream is really high because we found a lot of those group 1 organisms. The lake, still pretty clean water for New Jersey, but many of those organisms fell into that group 2 category. And so that would indicate that there's a lower oxygen content and maybe a slightly higher level of pollution at that lake. And then, so the big question is, why do we care about this stuff? Well, like I said, bugs can tell us the story of our landscape if we know how to listen. And a lot of research, including my own thesis research, has been focused on identifying these bugs and trying to figure out how people can use this in terms of research. And one of the great tools, which I'm going to talk about in just one second, is the Macro Blitz tool. And this information can help inform land management decisions, and people all over the world, if this data is accessible, can look at it and decide what they can do to help improve the quality of the water. So iNaturalist is the other project that I've been working on with National Geographic. If anyone has used this before, you know what a powerful tool it can be. There are about 3 million people now using this. It's an app that you download for your phone, or an application that can also be done through your computer. And it's an online social network of people that are sharing biodiversity information to help each other learn. And basically, the way that it works is that you go out into the field and you take a picture with your phone, and you can share information relating to what you're finding, what kinds of plants or animals or fungi. And artificial intelligence, or the computer vision model that's used by this app, can identify the organism. And then other people that are in the iNaturalist community will verify or suggest another identification for your observation. And the idea is that it's this really cool crowd-funded citizen science program, and all the information that's out there is accessible to people all over the world. And so it's a really cool way to connect with other people. I have uploaded observations of bugs that I didn't know anything about, and other people contacted me and said, hey, I'm doing research on that kind of insect, and you putting your observation up there has been really valuable data. We're also using it at the School of Conservation. So if you look, the project that we have going on now that encompasses all of our property boundaries has so far collected 3,181 observations, and we're coming really close to 1,000 species documented on our property. And again, this is plants, animals, and fungi. And all of these observations have been made by our staff or other visitors that have come along on our property. But it's a really wonderful way for us to start exploring and measuring the biodiversity that we have on our site. So this last thing that I want to talk about before I close tonight is the idea of this Macro Blitz project. So you learned a little bit about the aquatic insects, the aquatic macroinvertebrates that I'm really excited about. And now I'm part of this team with National Geographic to help launch this new project. And the idea is that we want people to start using that iNaturalist app to document the benthic macroinvertebrates that they're finding when they're out in the field. And you can do this too. So yesterday, when I was out making the video for your your virtual field trip, we found that giant hellgrammite, the one that was on the spoon, and it's also called an Eastern Dobson fly. And I uploaded that observation to iNaturalist yesterday. That's the screenshot that you're looking at there. And you can see that that observation has already been verified by somebody who's into bugs with a sense of humor. His screen name is Highway to Hellgrammite, and he verified that observation. And so now this is a really valuable piece of information because we know the date and time and the location for where it was collected, and it's been verified that it is that organism by someone else. Maria, do we have time? I have another 2-minute video, and I know the sound works on this one because we tested it together. Do you want me to skip that, or would you like me to wrap up?
No,
I know
sure,
we're a little...
continue.
Okay. So Dalal Hanna is one of the National Geographic explorers that I'm working with on the project to launch Macro Blitz. And this is a video that we made that has not been released yet, but it's a sizzle video that National Geographic will be releasing for Citizen Science Month. And Dalal is an entomologist working in Canada, and she's talking about her work and Macro Blitz. All right, everybody. So technology is obviously, I have bad technology juju tonight. I apologize for the delay there. Again, it's tested before we started. The last slide that I want to show you, if I can get out of full screen mode, is just your your challenge. So my homework to you is to get out and explore, and it can be any local body of water near where you live. It could be a pond or a stream or even just a trickle. You can download the iNaturalist app for free and take some kind of pictures of your discovery and upload them. And I promised that I would let you know about an app that's available. It's called Pocket Macros. Again, that's also free, and it's a wonderful key that you can use to help with identification. So thank you from all of us. And my very last slide has my email on it. I will also put that in the chat. So if anybody has any follow-up questions or wants to come out and look for bugs with me, I would be happy to go with you anytime.
Tanya, can you share your slide?
I sure can.
Yeah.
All right. Does anyone have any questions?
Hi, Tanya. It was a great presentation. I really was taken into that all the different microorganisms we can collect even from a small creek, right? Really love this idea of microblitz. I mean, I think it would be a very novelty to get our community engaged and trying to kind of give us more information about the diversity that we have here in our state. That's something I would love to find more information, and maybe I could share with the New Jersey Academy of Science network, so they can use it specifically. This is great for the classroom, for the teachers to kind of start using technology and actually get students engaged into a real research. Because I think imagine, like, you could use this in the classroom, and then somebody in South Jersey, for example, has seen the same animal, and you're like, okay, how big, how small? I mean, those are the things that we are observing, right? It's a great job. I'm looking forward to our collaboration in the future. I would love to go to your site now that I saw it by the videos. Oh, and hopefully we could coordinate our intent collaboration event that we're thinking about. Thank you
That sounds
so much.
great, Maria. And I am much better when I don't have to plug something in. So we will not have as many technology glitches when you come for a visit at the SOC, that's for sure. I see we have a question in the chat. Isha's asking if only schools can come to the School of Conservation. So no, not necessarily. We have lots of different user groups that come to visit us. Sometimes we do programs for scout groups. We have other types of schools other than public schools, like Montessori schools come. We're doing a lot more work with university partners. Generally, we don't do private events. We just do things that are focused on education and our mission. And, we'd certainly welcome you if you had a group like that wanted to come up and visit. Scott, you have a question.
Yes, thank you. Tanya, that was just wonderful, and it was so lovely to see you teach again. So it's been a while since I've seen you in action. And my question is kind of related to the teaching side of things. Do you have any, I'm sure there are some teachers and definitely students and some teachers online here. If I was a teacher and let's say I'm maybe in an urban school district and I wanted to do some things kind of like this, but I maybe don't have access, easily to come out to the beautiful locations that you're showing us. Which, by the way, everyone, if you've never been there, it is literally the most beautiful place in New Jersey. If you can get up there to work with Tanya, that's the best place to go. But if I can't, and I've got a school group, do you have any suggestions or maybe point people in the direction of maybe some online resources for like lesson plans and things for like urban ecology or, backyard ecology, like those kinds of things?
Sure. So that's a great question. And I have a two-part answer to that. The first part is I used to teach in Plainfield, which is a pretty urban part of New Jersey. My students and I had to walk pretty far to get to a stream, probably like 6 or 7 blocks through the city of Plainfield, but we made the trip and it was worth it because the students developed a real sense of place for their community. Many of them didn't even know the stream was there. They hadn't been there before. And so sometimes you can get access in more urban areas, but it is a little bit more of a challenge. If you can't get access, there's an online component to the iNaturalist app where you can download any data for your community. And, it may not be easy to get access to a stream, but I did a lot of education with my Plainfield kids in the parking lot of the school or the grocery store parking lot across the street. It was amazing how many great birds we found there, how many insects we found. Even if the aquatic ecosystem is a limiting factor for you as a classroom teacher, there's still a lot of nature outside all the time, no matter what kind of environment you're teaching in. So you can still go out and use the iNaturalist app, even if you're not in a stream. MacroBlitz would be a little tougher. But the cool thing about iNaturalist is you can download the data from anywhere in your community, and you can export, you can set parameters and look at the geographic boundaries and be specific about the kind of organism that you want to study. And you can actually download an Excel spreadsheet of all the observations that have been made in your community. And, if you look at that map of the iNaturalist scale and scope at this point, with that many people making observations globally, there's observations everywhere now. It's a really cool citizen science program that's really accessible to everyone. And the MacroBlitz website that I just put in the chat, we're putting a whole module together to help people understand how to use it. And there will be resources for classroom teachers there. We're also running a workshop at the School of Conservation this summer in August. And if teachers want to apply for that, they're welcome to do that too. And I'll put our web address in the chat for that as well. It looks like Maria asked a question. Can we use a puddle of water? It depends on if it's there long enough. You might be able to find some things in a puddle. We found mosquito larvae, they lay their eggs and go through their life cycle pretty quick. So if the puddle's big enough and it hangs around long enough, you might be able to find some life in there. That's a great question. Okay, any other questions? Oh, sorry.
Yeah, sorry. We usually used to get scared by seeing those insects because some might be poisonous or something like that. So do you put any vaccines or how to identify the poisonous insects or like?
So we don't have too many things that we have to worry about in New Jersey. I mean, mosquitoes can carry some kinds of diseases, but for the most part, the things that you're finding in the water in the larval or the nymph phase, you really don't have to have any health concerns about those things. So, I wear some bug spray on my body before I go out into the field if I'm worried about getting bit by things that are flying around. Typically, it's just a few mosquitoes, but I don't put anything on my hands when I'm handling the insects because obviously that would not be good for the specimens, and we want to return them to the water in the same good condition that we collected them in. But yeah, the level of risk is very low. Good question.
Yeah. Okay. Any more questions? Do you collect some insects in the lab and do any other kind of studies like breed or do something in the lab?
We haven't, but for my master's thesis, I collected almost 4,000 specimens and identified all of them. So I spent a lot of time looking at them through microscopes, and it was a lot of fun because you get to collect them in the field and then bring them back to the lab and really get a sense of, what the anatomical structure looks like, and you get a sense of, why certain animals live in certain places and have certain features like gills or little claws on the ends of their feet. And that part of it is really interesting to see it at that scale too.
Oh, understand. Any more questions?
Yeah. What about genotyping analysis? Like just to identify specifically, if there's any any changes, mutations that can be happening, now that we have molecular biology that can help us and guide us a little bit more.
That's a great question, Maria. I'm not sure, Scott. I don't know if you have more insight into, genetics of insects. That one's definitely a little bit out of my wheelhouse.
Yeah, I'm also a lot like Tanya. I'm more of a buckets and boots. As you were saying, you can do a lot of your stuff with your kitchen, and I can too. But I will say, I just sort of in a novice way of answering that question. I know that our colleagues, who do next gen sequencing, DNA sequencing, and these kinds of things, like they have very interesting. Again, I see some hands going up there. So very interesting techniques for doing like high throughput stuff where, like they're literally dragging the oceans to find new, genotypes that have never been found before. So it's like 20 years ago, these things were science fiction, and now these are tools that biodiversity specialists are using. So I think the answer is yes, but I probably Tanya and I aren't the best people to answer it.
Thank you.
Thank you so much. So it's wonderful talk. Insects have stories about landscape, which is very nice to hear from you, Tanya. Thank you so much for this wonderful talk. And I think students will be approaching you for summer programs or any kind of training. Definitely. Thank you so much. So let's move on to the Okay, so next we have, yes. So next we have some presentation from students. So first presentation will be from Mahika. So remember, this is not a competition. However, students will have only 10 minutes to present and 2 minutes for the questions. I would ask you to please mute yourself until the question and answer session of each presentation. I will let the students know when they have 2 minutes left in their 10 minutes presentation, as we have included many young innovators as possible in the time frame of the evening. Presenters, please stick to your time. And you have to share your own presentation and control your slides. So the first presenter will be Ms. Mahika Java. Ms. Mahika Java is here.
Yeah, I'm here. Thank you for having me.
Yeah, welcome Mahika Java, and your time starts. Can you just share it so that I can start the timer?
Yes, I will share it right now. All right, can everyone see my screen?
Yes.
All right, perfect. So, hi everyone. Today I'm going to talk to you about an initiative I started with my friend Lena, who wasn't able to make it today, but we started it in the 8th grade. But before that, I just wanted to thank Dr. Agapito for inviting me to present today. So in the summer of 8th grade, it was right when COVID was kind of coming to an end, and we realized that ourselves, we didn't really understand and like get the same experience out of all of our classes that we did through Zoom. And my friend Lena and we came up with this idea that whenever we did hands-on experiments at school, we definitely enjoyed it a lot more than the traditional form of lectures, like droning on for 55 minutes or a class period. And I would often find myself doodling on my notebook paper or just not really paying attention unless we were really doing something to interact with our hands. So definitely so we came up with the idea to implement this into especially neighborhoods and communities with schools that don't have the same budget and all of the same access to resources. So our idea was to create a summer STEM program. And this program would be completely teen-led, free of charge, and most importantly, hands-on and engaging. So in order to implement this idea, it took a lot of planning. So the first thing we needed was funding, of course. So along with our idea, we needed funding for how we could get these camps in action. Because as 8th graders, we didn't really know what to do, where to go. So we had a lot of mentors who were able to tell us how we could get these camps started and make our idea become a reality. So we got funding. We reached out to different life sciences companies, and we wrote grants, and it took a lot of effort to get our idea through to a company. But the first company that kind of gave us some recognition and wanted to help us kickstart this program was life sciences company Bristol Myers Squibb. And they even said that they would love to help us talk to the students and run experiments with us. So having a real scientist was super cool, and we could just see our idea coming to life in the program itself. This was our first year, so both my friend Lena and I were still in 8th grade, and we were just really excited to put our ideas into action. So the next thing we needed was a space, and that kind of tied together with the community support that we got. We reached out to a bunch of different community centers, youth centers, and really just all sorts of things, even schools, to see if we could get into a space that we could run these programs free of charge. Of course, some of them definitely said no, but others, for example, the Middletown Arts Center, said they would even give us the space to use for free, which was amazing for us as a newly running nonprofit in a sort. And this Middletown Arts Center also offered to help us advertise for our first round of camps ever. So just getting community support, she helped us, the woman who helped us get into the space helped us advertise our program to students in the community. So next we had to implement it. We had our space, we had kids, and we had funding, we had our supplies. So the implementation, so far in the past 3 years of this program, we've done it at the Middletown Arts Center, which is more located in Central Jersey, the 2nd Street Youth Center, which is in South Plainsfield, New Jersey, and in the Wall Municipal Building, because we did that one with some of the Girl Scouts, because one big part of our program is also empowering girls. As 2 teen girls who want to see more younger girls getting more interested in STEM, we wanted to propagate that more. So you can see here some of our scientists who have came in and worked with the students. And so now we go into our impact. So you can see here a lot of our students really enjoyed our camps. The experiments that we do, we make sure that we keep a good complex scientific level while also making it fun and exciting. So of course we have to do our small volcano experiment with baking soda and vinegar. And we also do what's a fan favorite is the mucus experiment or the allergens experiment, where we make slime and simulate how our nose creates boogers and other allergens. So this is an example of a video of an experiment that we do. So that's just a small clip of the video, and it was of extracting DNA from an onion. But beyond our local impact in New Jersey, we were also able to reach out to other countries, and through posting our program on LinkedIn and other social media, showing what we were doing, the camps we were running as younger students, teenagers. And this went into helping fundraise for a menstrual hygiene in Uganda. And we were able to give a seminar on the scientific health and how to maintain good hygiene during your period. So that was one way we extended our impact globally. Another way we extended our impact globally was to India, where we would we do webinars almost every Sunday with students in underserved communities and slums in India, where we would present the same topics, but through a Zoom platform. So you can see here I was presenting this parts of a cell and a nucleus and the DNA, our DNA extraction experiment. I was also able to go and run these experiments with students in India when I went recently, at the beginning of this year, and we were able to reach out to a school with all girls and show them how we can do hands-on experiments and actually apply all the science we learn into and see how we would actually use that. So in addition to STEM powering, I also am the president of the STEAM Park Teen Board, which is in New Jersey, and it has a similar background, except it's much more established in New Jersey, and it runs year round. And we not only mentor younger peers, we help lead community events like fundraisers and reach out to students and families around New Jersey. So some things that we do are this enrichment program, STEM Girls online weekly workshops. And so definitely reach out to me if you're interested in volunteering as a teen or even an adult. We'd love to have you. So after 3 years of STEM powering, we hope to continue on with this trend of reaching out to underserved communities in New Jersey, and we hope you can join us and volunteer for some of our camps. Thank you.
Wonderful. You have 20 seconds left on time. Thank you so much for the wonderful presentations. I'm so impressed by the work which you're doing to the community at this age. So it set an example for all the youngsters. So any questions from audience? Yes, Tanya has a question.
So amazing work. I think you all have done things that many adults who have tried to create similar programs have a hard time doing. So congratulations. And I think it's so great that at your age, you're able to accomplish this much. What are you hoping to do going forward? Are you thinking about a specific career path in college? Do you want to do science education? I'm curious about what your long-term goals are.
So, well, I've always been interested in engineering, but I definitely want to tie in like some aspect of education going into college. I'm not really 100% sure yet. I'm still a junior, but I'm really thinking about like tying in that aspect of education and business into like a STEM major. So that's something that I'm interested in doing. Yeah.
That's awesome. And Maya, I have one more question. What's your biggest challenge been in putting this project together?
So definitely getting volunteers, because often students show the interest, but when it actually comes down to the time, we need volunteers, and that's also a struggle because people, maybe it's too far away or they don't have a ride. So it's often a struggle, like getting people interested in the topic and the program itself. So yeah.
Thanks.
Yeah, thank you.
Tanya, you actually stole my question, so I had to think of something else because I was going to ask the same thing. Do you want to be a teacher? Because I think you would be a wonderful one, and I hope that on some level you consider that, you know, as part of your future, no matter how that might be, to educate others. What you showed us was incredibly inspiring. And I got to be honest with you, you know, I'm a college professor, and you run circles around a lot of college professors, and the impact you've had on other people. The presentation you gave was so well done, and you hit all the things that, again, somebody like me would do if I was talking about something like this, and better actually. So So congratulations to you on this really amazing work. The question I have for you was to ask you about your mentors. So you talked about you and your friend and how you did this work. Did teachers help with this, or who were sort of your mentors in putting this all together?
So we had a lot of help from our community, as I mentioned. So definitely a lot of the scientists from BMS actually met with us multiple times and like kind of led us through how we would run some of the experiments, helped us plan experiments. So people like even my teachers as well. I asked one of my engineering teachers at school to help me design an experiment for my students, and he was more than happy to help me simplify down an experiment. He helped me design our paper airplanes experiment. So we really go into that as well. So definitely we used a lot of our community to help us.
That's wonderful, and I just want to, say any student like you, whoever reached out to me for help and mentoring on something like this, I would have been, overjoyed to be able to help. So I'm glad you've got folks like that in your support network. Well done.
Thank you.
If you have a video about what you're doing, and if you have any flyer, please do share with me. I have so many WhatsApp community groups with lots and lots of family. I can share with them. Definitely, you will get lots of volunteers. And what you're doing is so amazing. And we can also share in our annual meeting, right, Dr. Maria? You can share your video also in our annual meeting, which is going to come on April 20th, where 188 youngsters, junior academy students will be presenting their work. So it will be very useful for you also to elaborate your comment. Any questions? Any other questions? Okay.
Can I say it was a
Yeah.
great Mahika. It was very good. I'm very proud of you, and I am looking forward to a great event on April 20th, and I'm very pleased you will be joining us there in person.
Thank you. Yeah. I'm happy to join, happy to present and be there.
Thank you so much, Mahika.
Yeah, thank you for having me.
Thank you. So our next presenter will be Miss Divya Krishnan is our next presenter. Miss Divya Krishnan is here. And she will be presenting on utilizing AI algorithms to classify space debris from Edison High School. Yeah. So stick to your time. When you're ready, I can start the timer. Your timer starts now.
Thank you so much for having me present, and it's an honor to be here and also hear the amazing presentations I've heard so far. So my name is Divya, and this is my project using AI algorithms to classify space debris. And so now let's get into what the problem is. So if you think about space debris, we think about just old satellites or maybe some old rockets that are roaming in our atmosphere or even roaming in low Earth orbits or many high Earth orbits. And we can't really conceptualize what exactly this is because isn't really covered in a lot of space news. But if we think of just a simple paper clip, a paper clip you use for school, for work, for everyday use, think about how small that is. You can barely fit it on some people's index finger. But these paperclip-sized debris can travel over 100 miles per hour. And with this paperclip, there are over 128 million of these paperclips constantly roaming our low Earth and high Earth orbits, posing an extreme danger for so many different spacecrafts as well as satellites constantly orbiting our Earth. And not only does this result in billions of dollars worth of damage, but it's very dangerous. Say we wanted to launch a spacecraft into orbit. It would be extremely dangerous to have this amount of space debris constantly roaming our orbits. And because of the sheer amount of such debris, it's extremely difficult to track. And me personally, I'm extremely passionate about this problem, and especially anything to do with space. And as you can see here, the accumulation of space debris over the years has been astronomical, and detecting such debris has been more and more difficult. So before we get into the research, why exactly does this project kind of make a difference, and why is it different than any other old research project? Well, because you have so much data, amounts of like petabytes and so much that are constantly coming into Earth, it's very difficult to be able to classify it accurately, and many current research uses just convolutional neural networks, which are networks that are able to detect images, but usually only one class or one object at a time. While this research uses a YOLO V8 algorithm, which I'll get into a bit later, that is able to classify multiple different objects, and especially at different conditions and varying conditions as well. And with this research, I do use synthetic data. However, this data is modeled after actual space conditions, including using a lot of background noise, which spacecrafts usually in orbits encounter. So my hypothesis was, given the immense amount of space debris, using an YOLO V8 algorithm applied to data with realistic conditions leads to accurate and greater classification of data. So now, what exactly is this data I have been talking about? So this was synthetically created by the University of Luxembourg, who was able to reach out to an email because I really wanted to do this project, and a lot of data, as I said before, was very hard to collect. So here are some images of the data set that I used. There were over 100,000 images in the original data set, ranging from small pieces, as we can see here, that are extremely low brightness, to huge satellites that are orbiting Earth with very high brightness, as in this image right here. And so again, it includes like those paper clip sizes from 1 centimeter in diameter all the way up to tons of huge space satellites that are modeled after actual space satellites. So how does this algorithm work? So we have an input, so this would be our image, and what it's able to do is it's able to take this image, and each pixel, it's able to split it up into a pixel grid. And with each pixel, we're able to put a bounding box over it. And a bounding box is just a box around your image that you're wanting to classify, or the object you're trying to classify, and it's telling you, is it this or this? For example, if I had a cat and dog in an image, it would tell me a bounding box would be over that cat and dog, and it would be, is this a cat or is it a dog? And so it's able to input and output confidence values of whether it's one class or another. And in my case, because I had 11 different classes, it would give its confidence on these 11 different classes, and then make a final prediction based on the various features it's able to break down from all of these different pixels. So with my methods, because the original data set was not in the YOLO V8 format, I had to redo the labeling for that. And so I first used an annotation software called RoboFlow, where I was able to individually annotate the images, and then I was able to create aggregates of these different classes and multiply them based on the amount I wanted in my data set. And then it was split into a 80% trained, 20% test split, with the model application being YOLO V8, in order to balance the data set to reduce any sort of bias that could be introduced into the model. I was able to undersample, meaning the class or the specific amount of images that are the lowest, every single other preceding class would be lowered to that. So for example, with my data, there are 100,000 different images, with 15,000 being in the lowest class. So I was able, or not 15,000, 5,000, and I was able to reduce that to 55,000 total images. And so I was able to split the data into various groups, and these groups included brightness as well as the size of an object. So with the brightness, I split it into dark, medium, and bright groups. And here's kind of the accuracy between each of the groups, with blue being dark, pink being medium, and yellow being bright. So essentially what this means is I had 3 different sort of groups that had these 11 different classes in each one of these groups, and it was able to classify these different varying levels of brightness with the yellow V8 algorithm. And clearly, no matter what the brightness of the object was, it was able to classify it very well and correctly. And then sizing, I varied the sizing. So as I said, because there are very small objects to extremely large objects, it was a bit more variable in classifying the objects and a bit better in classifying the various levels of brightness. And so how are these results applicable? Well, with constant data coming from sky surveys, telescopes, rovers, everything you can think of in our orbit, it's a data firehouse. We have to be able to take in this data, really conceptualize it, understand what's happening, and be able to classify all of these hundreds and millions of debris floating in our orbits. This is also an extensible solution able to be used by Air Force to further classify objects and use them in prevention methods. And tracking and cataloging space objects is critical to avoiding accidental collisions, defending against adversarial activities in space. And then money. Money is one of the biggest factors in why a lot of companies are interested in this research, as it saves them billions of dollars in funding as well as repairs that could potentially be caused by all of this debris. Thank you so much for your time, and I hope you enjoyed listening.
Thank you so much, Ms. Divya. You're also on time. You have still 1 more minute to complete. Great. So it's a wonderful presentation. So any questions from audience? So how did, yes, go ahead.
Okay. That was also a very wonderful talk, Divya. That was really, really good. I have a couple of questions and a quick comment. Do you think that if we were looking back, at the early space programs, in the 1960s, they wouldn't have been contending with as much debris? Is that correct? This micro debris, because we weren't putting stuff really up there yet, right? How would you characterize the sort of the difference between, like if you're putting people into space, people into space now, spacewalkers at this, International Space Station, like, are they facing a much more dangerous job in these kinds of ways than maybe space explorers in the past wouldn't have?
Yeah, definitely. So, essentially in the 1950s and 60s, because we weren't putting as many spacecrafts and you didn't really have as much debris, it was definitely much easier in that regard. Obviously, they had different challenges regarding the facilities that they had, and the technology that they had was more difficult to sustain these people. But now we have that technology, that ability to have these people go into space, but now we obviously face different issues. We have the accumulation of so much junk, and you would never think, we always think about on Earth, oh, let's focus on getting rid of the trash and everything, we can just go to space. But space is also starting to be crowded. Space is also receiving as much junk and as much debris as we see on Earth, and is definitely a challenge for people at the ISS. They constantly deal with it every day. Now they're making a lot of new technology to deal with that space debris, obviously with material science and the ability to create materials that can deflect it, but still it poses a very strong danger now.
Thank you. That's a very good answer. And I have a follow-up question, which is a little bit different kind of question. So it's kind of like I asked the student who went right before you, sort of what do you want, well, actually Tanya asked that question, but I was going to ask, what do you want to do when you grow up? And here's what I'm thinking is I have colleagues in, for example, the Department of Computer Science, and they study YOLO V8 and they use it for like machine learning and cars that can drive themselves. So they're recognizing, people from dogs, from fire hydrants, and then I've got colleagues who do, very deep space research in the Department of Physics. And so your work here is kind of a blend of lots of interesting things, bringing kind of computer science, data science, space science together. So what do you want to do someday?
So, I really want, like, ever since like literally second grade, like aerospace engineering and like that has always been kind of my passion. I want to work more on the software side of it, obviously, but definitely being able to like build and like being part of that space community is something that I've always wanted to do, and not only work for like private companies, but be able to really help the human race, like especially in the future when we do look to space for materials like mining. I've looked into space mining as well as establishing habitats and even finding life on different planets. Like research areas like that are stuff that I'm like really interested in, and making those like crafts that go to these places are like my passion.
That is so exciting. I wish you the very best. I know you're going to do and discover very amazing things in your career. So best wishes to you. Good job tonight. That was very well done.
Thank you.
So one
Great
question, a
job.
quick question. So how did you end up in space debris? What inspired you to study that? Just curious.
Yeah, definitely. So, basically, I do a lot of personal satellite tracking on my own. It's definitely a science in and of itself. And one day I saw like one of the most like significant satellites that the European Space Agency used actually was hit by a piece of space debris and fell into the North Pacific Ocean. And this cost them billions of dollars, as well as a lot of implications because it dealt with climate tracking for the Europe, and it was very detrimental to many um, going on activities throughout that whole month that they had to try and get that satellite back up. And that first inspired me to think, well, there aren't a lot of prevention methods to actually deal with something like this. And I was able to attend a keynote talk because I love attending like anything that has to do with space. And it was part of the Aerospace Corporation, and he was working on actually detecting space debris. And he kept on saying like, because there's so much, it's so difficult, because there's so much, it's so difficult. And after that, I was like, I need to be able to solve like some sort of problem like this and like apply my knowledge. So yeah.
This is awesome. I think Dr. Maria has some question.
Yes. Hi, Divya. I am, wow, this is great job. You have raised very important points from political economics levels, just as Dr. Kite mentioned. I'm very proud of you that you're taking to the next step, and very fortunate to know that the youth is actually already thinking ahead of the things that we need to be, that you guys are going to be facing in the years to come, because space debris, space innovations is the way we're going. It seems that next 2030 will be the whole space innovation, and we are already seeing that. So I hope you could join us on April 20th, and see you there in person. Please make sure that we get a chance to meet.
Definitely. Thank you so
So
much.
much time.
Thank you so much, Livia. And our next presenter is Mr. Gunavan Kezia. Are you there? Oh, sorry. Miss Gunavan Kezia. Go ahead. You can share your screen. Can you hear me? You're not.
Sorry, it says that the host
You
disabled.
got it. Yes. Can you please share now?
It says that it's disabled. Oh, okay, no, never mind. It went through. Hello, my name is Keisha Gunawan, and I'm from Westwood Regional High School. And first off, I would like to say that thank you for this wonderful opportunity that you have given me, and that I'm so thankful to be here. And my presentation is going to be about the ultrasensitive magnetic trap tiltmeter for volcanic monitoring. This is my outline, which consists of the introduction, my problem, objective, and hypothesis, my methods and backgrounds, and my materials and construction, my results and discussions, my conclusion, and references I'll be talking about in this presentation. First off, I'll be talking about my introduction, where I will talk about volcanic monitoring and how I'll be using the tiltmeter to help with it. As we all know, millions of people around the world are living in volcanic hazard areas, and there's a such case as in May 1980, where Mount St. Helens in Washington state erupted, and it is considered as the most destructive eruption in the US history. And this specific event, scientists have shown that the ground tilt changed slowly during a few months before the eruption, and it also accelerated right before the eruption too. As you can see here on our, as you can see here on our graph of the tilt angle as a function of time, there's a very small change in tilt angle of 0.15 degrees right before the eruption, which is like a few weeks just before the eruption. And next I'll be talking about the tiltmeter and what it's about. And the tiltmeter is a device to measure the inclination angle of a surface, as we all know, and it's also used in many applications. For example, home and building constructions, public structures monitoring such as bridges and dams, natural hazard monitoring such as volcanoes and landslides. And some examples of the tiltmeters are the bubble tiltmeter, which is common and cheap, which is usually less than $10, but it's not as precise. And the next tiltmeter is the electronic tiltmeter, which is precise but pretty expensive, which ranges from about $100 to $1,000. And next I'll be discussing my problems, objective, and hypothesis. And my problem is that in many of tiltmeter applications, we need sensors that have high accuracy, but also a low cost, so it'll be okay for it to be destroyed by the volcano. And the common bubble tiltmeter is not sufficient for this, while the electronic tiltmeter that we have now is very expensive. And our objective is simply just to create a high accuracy and low cost tiltmeter, so it could monitor volcanoes and also other natural disasters and public safety applications, for example, monitoring bridges and dams. My hypothesis is that there could be other methods to detect the ground tilt with better precision and also a low cost. A magnetic levitation device, such as the one that I'll be using in my project, can also be used for this purpose and could be because it has no contact. We can also use Raspberry Pi and a camera for automatic electronic detection of the tilt. My methods and operating principle. For my method, I'm going to be using the diametric levitation, and in my project, we're going to harness the diametric levitation effect to create a tiltmeter. So a diamagnetic material is a material that is repelled by magnets, and in this case, we're going to use a pencil lead. And for here, I use a special arrangement of two diametric magnets called the parallel dipole line, or the PDL trap, where it could levitate and trap a piece of graphite in the center, as we can see here on this diagram. These are the cylindrical magnets, and this is the south and north pole as indicated over here. And this is the magnetic field in the center, which will hold the graphite afloat. Next, I'll be talking about the camelback effect from the magnetic trap. And our PDL trap has another interesting behavior, which is how the graphite levitates and is stable at the center. And this happens because the magnetic field is slightly stronger at both sides. And the plot looks like a camelback, and inside the middle, the magnetic field is a little weaker than the edges. And this effect gives stability to the graphite rod and is often called the camelback effect because of its structure. This phenomenon is similar to a ball inside a bowl, as it can oscillate and slosh back and forth as the center. And my observation is that if we tilt the magnet just a little bit, the graphite moves with significant distance. Thus, it can be used as a very sensitive tiltmeter. This is my system construction, where this is the box frame that holds the structure together. And this is the camera that will be taking the pictures of our PDL trap here. And then we'll have our Raspberry Pi microcomputer that we'll be using. And then we'll have the magnetic trap and graphite, which is the heart of our system. And on the bottom, this white platform here is a test tilt platform, which is controlled by the turn knob, which can measure, like the tilt angle can be changed by 1 turn, which is 0.1 degree change. Next is I'll be explaining my tilt detection program. And to start, we'll capture the image. And just to clarify that the image is taken in the grayscale so that when we take the next step is taking the cross-section line, which is this red line that's indicated over here. So it'll be easier for the computer to see the value between like where the red line is scanning. So over here, we can see that it's black, but then since the lead has some sort of shiny exterior, the computer senses that, and when we plot the line scan as seen over here, this is what we see as the pencil lead. And then we'll find the edge crossing by, through this, we'll have the threshold, which is represented as the green line. And then the zero crossing is the red dot over here. And then from here on, we can calculate the tilt angle using the calibration data. And then we'll repeat this process until we see a significant change, and then we'll stop there. And then I use Python to program this Raspberry Pi, and I use several important programming concepts, which to start with, I learned how to do numbers, string, list, tuple, and dictionary. I also learned how loop control works and camera control, image detection, image and data plotting, and zero crossing detection. These are my results in discussion where I tested my tilt meter. When I tested my tilt meter, I actually performed calibration by removing the graphite edge from my program and increased the tilt in 0.1 degree steps. My test actually shows my tilt meter has several excellent good characteristics. It has a good range of negative 0.7 to 0.7 degrees. And as I said before, the tilt angle in Mount St. Helens was only 0.15 degrees. So this is a very good range since it'll have it's a larger range. And secondly, there's a high sensitivity between negative 0.2 and 0.2 degrees, as we can see here, because it's a smaller slope. It can also detect a very fine change, which is has a high resolution as low as 5.4 times 10 to the negative 4 degree over pixel. And therefore, my tilt meter is very sensitive. It also has a good range and is low cost, which will make it suitable for volcanic monitoring. Finally, in my conclusion, I have learned about diamagnetic levitation using 2 diametric cylindrical magnets and a graphite rod in the middle. The camelback effect and the parallel dipole line magnet trap causes the graphite to be stable in the center. This trap allows us to build a very sensitive tilt meter with a range of plus and negative 0.7 degrees and a resolution as low as 5 times 10 to the negative 4 power degree over pixel. Using the Raspberry Pi and Python, I have built a very low-cost digital tilt meter, and which will be very useful for monitoring volcanoes and safety for many public structures, such as buildings, dams, and bridges. And that's it. Thank you for listening, and let me know if you have any questions.
Thank you so much. You're also on time. You have still 1 more minute and 27 seconds. Great. So do we have any questions for this wonderful presentation? Okay, so where do you use this device actually? Like for, where do you keep this device to sense the volcanoes?
We will keep the device. So I have a model of like a volcano actually here. So we will keep the devices on like different points, such as like on top here, and then on the edges here. So like we can like detect the tilt angle from like the top.
So how did you get this idea of studying volcanoes?
So initially we actually started with we had this project a few years ago back in COVID where we actually started with just a tilt meter, and using the PDL trap. And then we figured out that like we could use it for many more, we can apply it to like natural disasters, and which will help with like people around the world who are living in these like endangered places.
Wonderful. Thank you. And Scott has a question.
Yes, thank you. Kezia, that was also, we've got some really good speakers tonight. That was so well done. And a couple of comments and a question. I was about your age when Mount St. Helens erupted, so I remember that happening. But I was commenting to a family member who's sitting off camera. I was like, when I was that age, a science project for me was, collect leaves from 20 different species of tree. It wasn't, invent a gizmo that can, measure tilts and volcanism and predict eruption. Like that's pretty amazing. And a lot of stuff went into your project. You were doing some physics, you were doing some computer science, you were doing some engineering. And I'm just like, wow, so deeply impressed. And so my question is, I learned something tonight. I sort of, like most people, have seen little toys where something appears to float, because of the magnets and things like that. But you identified this as a really interesting application of this, and you've sort of designed this sensitive instrument that doesn't have the problem of friction. And I was just wondering if you had any thoughts, like, was that part of what, led you to choose this particular way of solving this problem, that you don't have to worry about friction? Because I was thinking, you showed some pictures of the bubble tilt meters we use for like carpentry. You want to put a board on straight, use the little bubble meter, but sometimes that little bubble gets stuck on the glass, right? So your contraption doesn't have that problem. So I just wondered if you had a few words to say about that.
Yes, actually, we did think about that, and how since it was the magnetic like field, it doesn't have any like some sort of friction that will act upon it, so it'll be more accurate than the bubble tilt meter. And also considering that like the electronic tilt meter, even though it is very accurate, it's very expensive, so it'll be like a waste of money if it gets destroyed by the volcano.
What was the, if you had to guesstimate, because this is a prototype, so if you were going to mass produce something like this, it's going to be even cheaper. But like, what was your kind of total cost for putting this thing together?
About like $100, like around that amount.
Wow, very, very good. Well done. That was really cool.
Thank you.
Thank you so much, Kezia. Any more questions?
I would like
It's
to
such
a comment.
Yeah.
And very good work, Kessia. I'm impressed. I mean, we are seeing, with those, with what we're seeing with global climate change, right? We also seeing disruptive movements now in different volcanoes around our planet that are becoming active in the ability to foretell this in advance. It will be something that could be very helpful to many people that live near these entities. So thank you so much for your presentation. And once again, maybe when I meet you, maybe will you be coming to April 29th event? I would love to see your prototype in person and meet you in person. This is great. This is great work.
Yeah.
We will have our group meet during lunchtime, and I'm hoping that maybe you could discuss with peers your prototype. It would be a great event, so I hope to see you there.
Thank you. I'm looking forward to see you too.
Thank you so much, Kezia.
Thank you.
And our next presenter is Isha Vanamala. Isha is there? Yes.
Hi.
Go ahead.
I just want to say thank you again for the honor of presenting today. And I will just pull my it says host disabled. Okay. It says host disabled participant.
It should be ready to go.
Now you are the host.
Okay. So hi, as I said, my name is Ishanamala, and my project today is called Nocturne Insight. So a brief intro into my project. It's a sleep mask for accurate sleep classification and duration determination by using electrooculography and inertial measurement units. And I aim to extend the functionality by recognizing common sleep disorders and providing notifications to the user. My further developments would include a accompanying mobile app to interact with users and provide information on nearby sleep experts. Right now, I'm thinking of incorporating an alarm system that features a blue-enriched LED light to combat drowsiness and a speaker that allows users to fall asleep to customize soothing sounds or wake up to preferred tones. And now blue light can actually be used for something good because normally the blue light in your devices is the thing that makes you stay up late at night. So I'd like to go into some definitions and some basic concepts to fully showcase the depth of this project. And so EOG is short for electrooculography, and it's a technique for measuring the electrical potential between the front and the back of the eye. It can be used to diagnose eye diseases and record eye movements. An IMU is an inertial measurement unit, and it's a device that measures the force, angular rate, and orientation of a body, and it utilizes accelerometers, gyroscopes, and magnetometers. It can be used for navigation, motion tracking, and control of devices in this case. A PSG is short for polysomnography, and it's a type of sleep study that measures various body functions during sleep. And the combination of all these sensors together provides a really comprehensive overview of what stage of sleep someone is in, and it helps diagnose or rule out sleep disorders. Sleep stages, the 4 basic stages of sleep that a person cycles through throughout the night. The 1 REM stage, when you're dreaming and your eye rapidly moves back and forth, and your 3 NREM stages. So, number 1, the basics. According to the World Health Organization, about 1/3 of the world's population has some form of a sleep disorder, such as insomnia, sleep apnea, or narcolepsy. These disorders impair cognitive, emotional, and physical functioning and can increase the risk of chronic diseases such as cardiovascular disease, diabetes, and depression. I have had trouble sleeping for as long as I can remember, and it can be really discouraging to have someone tell you to just close your eyes and try to sleep or like count sheep, when in reality that's not the best advice for you. And having a device that's easily accessible to back you up on what you're thinking can be extremely self-affirming as a patient. Additionally, polysomnography electrodes can be extremely uncomfortable on different types of hair, so it may not reflect how a person would naturally sleep. So the materials that I used was an Arduino Nano 33 BLE, a computer with the capability to run Python IDE and Arduino, cloth, reusable electrodes, and assorted wires and connections. The main research question that I had starting out was how could a cheap sleep mask with EOG and IMU sensors accurately measure sleep stages and duration, and how would it compare to an actual polysomnography test? Objectives. So my goal with this was just to provide a sleep mask that could accurately and conveniently monitor sleep from the comfort of someone's own home. So here are my physical materials. Over here is the connections that I made on a breadboard. Over here is a Arduino 8C8232. It's traditionally used to measure EKG, but it also works for filtering from the signals from an EOG electrode. So I repurposed it for that purpose. And over here is the Arduino Nano 33 BLE. As you can see, it's extremely tiny, which helps to fit everything into the eye mask over here. And this part would be put over someone's eyes. So normally the person could do all of their daily tasks or their sleep routine before they fall asleep in this mask. And then when the sleep mask goes over the Velcro, the connection is fulfilled and the sleep mask will automatically start tracking. For my online materials, this was the website that I planned to make that would allow a person to train their own model based on how their eyes react to different types of sleep stages. And here were just some code snippets. Analysis. The code classifies sleep into one of 5 categories: awake, light sleep, non-REM, REM, and arousal. I combined the data from both the EOG and the IMU to determine this output. The code also recognizes arousal as awake-like movements for a brief period about every 10 minutes. This time is not attributed to awake, and by using the machine learning model created by the user on the website, the different numbers recorded from the electrodes are classified over the night. Any additional perks that the user wants, such as sleep music or awake music, can be set up through the future app, because the LED and the alarm are always set up in the Arduino. The mask fit had at least a 95% accuracy in detecting sleep stage with the mask. This is because on the website, a recurrent neural network is trained with data given by the user. More layers are added until the model reaches at least a 95% accuracy. The data given by the user is split into 70-20-10 for training, validation, and testing, and an RNN was used because the data was sequential and it was split with commas for 3-second intervals. I did test with an SVC, LSTM, and CNN, but the RNN was the one with the highest accuracy. This was much better than the performance of IMU and heart-rate sleep trackers that counted sitting still as sleeping. And additionally, preliminary sleeping tests where the user was videotaped so that they could later on point out the parts where they were awake and went asleep went very similar to the results of the sleep mask as compared to what Apple Health and different sleep tracking watches showed. So basically, here's a flowchart. The Arduino collects data from the user trains the model with the data, the model classifies as the person sleeps, the person is woken up, and there's now more accurate measurement of the person's sleep. Graphs and general conclusions. My project was really cheap. It's around $20 as compared to polysomnography tests, which cost an average of $3,000. And it did show that EOGs can accurately classify sleep stages and duration. There are some possible improvements on this, and I hope that it would inspire more curiosity into the role of sleep and how it plays into our lives. There is also some limitations. During testing, the user had to stay plugged into the computer, and in the future, when the Bluetooth function is truly added in, the sleep mask could completely be seamless. And another limitation that I have was that I didn't have access to a polysomnography machine, which means that I couldn't compare the results of my sleep mask with polysomnography. And although I can derive from the results that REM is noticed when the user woke up in the middle of a dream, this happens so infrequently that I don't believe that a high accuracy can be generalized for all REM yet. Here are my references, and thank you for your time.
Wow, thank you so much for this wonderful presentation. Yeah, usually whenever we don't get sleep, everybody says, just close your eyes. That never works. Wonderful. So we have a question from, yes, Dr. Maria.
Hi, Isha. I was very impressed. I must say, and I don't know if everyone knows, Isha is a middle school student, not a high school, middle school. Okay. Oh wow, I'm impressed. So I have I saw your model, and very interesting how you had it. Yes, you're right. 3,000 versus almost $20. That's a no-brainer. What? Question. I'm thinking things to consider. And I know when I saw it, the cloth was white. That could change. Do we have to consider the texture of the mass based on color or texture of the cloth? Would that matter? I mean, as you're getting into that REM sleep?
I would assume that a more silky texture would be better for being applicable for different skin types. And I know some of my friends have atopic dermatitis, and I wouldn't want to inflame their skin either. So having something that would adhere well on the skin and be stay on their skin and not either stick to their skin either and cause a bacterial buildup. So definitely the type of cloth used would be really important to make sure that the person has the most optimal experience.
And when we collected the software, and usually in a brain activity machine, you usually you're trying to, what was that software you use in order to allow the subject or the experimenter to know what are these values? What was that app that you use?
So usually they would the best way to do this would be with an EEG, which would be an electroencephalogram, and that would measure brain waves, and that would be much more better to measure the electrical output of the brain. However, since I was using EOG, there's a chip made by Arduino for EKG waves, and since it was so similar, I was able to just use that same chip, and it worked out just fine.
In what ways were you, can you retry? What ways were you able to get from that? What was it? IOD, right? What did?
I just used the Arduino IDE, and it measured the electrical potential.
Okay.
Yeah.
Okay. And how do you? I'm trying to think. So you measure the actual potential. So how do you know? How were you able to define the REM sleep then?
Okay, so when a person goes on the website, they do different types of eye movements. So the first thing they do is they move their eyes rapidly back and forth, and that helps generate some data values for that specific movement. And then they'd either slowly move their eyes and then like look straight and do like different tasks with their eyes like that. And then the model would train, and then based on that, their sleep would be when they sleep, they would then test through and then make sure the first night and preliminary test to make sure that when like they know that when they sleep, it says they're sleeping and stuff like that. But the model itself is what makes sure that they have the best sleep possible, and it validates them on a more objective outlook on, yes, they legitimately need help in having the best sleep possible.
Thank you.
Yes, Scott has a question. Go ahead.
Thank you. Me again. Isha, that was lovely. Hi. So I first of all, it's not so much about the science, but I want to congratulate you on a very well-tuned sense of comic timing. I literally laughed out loud when you went graphs, and then you went to the next slide. I thought that was very funny.
Thank you.
Yeah, that was really cool. I want to say that one thing I'm noticing tonight, and certainly in your work and some of the other student work, is how comfortable you guys are with programming and computing. That to you, it's almost like the afterthought, you know. So for someone like me, you know, like you were like, well, here's some code I wrote, and then you moved on. You know, for someone like me, that would have been the project. Like, oh, look at me, I wrote some code and it took me a week, right? And you're like, yeah, I just wrote some code to do this other more interesting thing. So that's it, just the toolkit you have to do this interesting work it's really impressive and something. I have a couple of questions, but my first question is you know, I do wear an Apple Watch and I wear it to bed, and so some days, you know, it gives me a hard time because I'm not sleeping well apparently, and then other days it's like, good job, buddy, you know, good sleeping, right? But from your research, it sounds like maybe my Apple Watch isn't as good at that as it thinks it is. And what are your thoughts on that?
Okay, so I actually use a Fitbit, and on days where like I make sure I have 8 hours of sleep, it tells me I have 6 hours. And I know that can't be true because I act like I make sure that I sleep for 8 hours because I want to be well rested for the next day. And something that I've seen is that since these rely on like just your heart rate and just IMU and how you're moving throughout the night, like you just sitting still and like staring at the wall can make it seem as if that you're sleeping when in reality you're not. Like your thoughts are racing, but well, I guess you're just calmly looking at your surroundings, so your heart rate is also lowered, but you're still awake. You're not gaining the benefit of being in deep sleep. And even if you're in deep sleep, like if you're dreaming and your heart rate's like extremely agitated, that could be like counted as being awake if the watch itself is not in tune with how your sleep stages go, like from like with your heart rate goes up and down throughout the night.
And then another follow-up, just another question I've been asking some of the other student speakers this question. What do you want to do someday? Are you interested in sleep kinds of things or engineering kinds of things, or what do you want to do?
So I'm really interested in anything to do with the human body. I think it's just such a marvelous thing that we exist the way we do, and almost everything that we have is made and it works, and we don't know why it works, but it works, and everything serves a purpose. We can heal ourselves. Like I find that so beautiful, and like the inherent beauty in nature is just it's I get awestruck just thinking about it every single time. So I guess doing something with the human body in the future would be a dream. And I guess like applying other fields and like forging a connection between them, like that's like amazing because you get to take stuff from other things and apply them to something and then make a totally new thing and advance those fields. Like that's beautiful.
It is, and I am so inspired by what you just said, that you have this sense of just life is so profoundly complicated and crazy. You know, we barely understand it, and I want to know more. I wish you well in your future studies. I know you're going to discover great things, and I know Mike has a question. I'm going to leave you with this. There's a famous developmental biologist who said something that sounds like what you just said. You know, imagine this first cell, right? The single fertilized egg from which you developed, right? Somehow that egg began dividing and differentiating, and it became somehow you. He said that is such a profound mystery that we should all be walking around in the streets proclaiming it to one another about how unbelievable that is, right? And so you have that, and that's just wonderful. And so I wish you the best. Good luck in everything.
Oh, in my one of my classrooms at school, there's like this pamphlet. It's like the mathematical ways to create you, and it's like, and it goes with like the powers of two, right? So then you have to have like 1024 great grandparents or something, and that's like like all those people made you. So like, yay, that's human life for you.
Yeah, Mike, go ahead.
Hi, Isha. Thanks for sharing with us tonight. I just have one quick question for you. How many different subjects did you test your device on?
So in order to comply with RB, I only was able to test it on myself.
Okay,
I would have to like go through that and in order to like test on more human subjects.
great. That was going to be my follow-up question. Well, best of luck. I really enjoyed your presentation.
Thank you.
Hi, Isha. I hope we get to meet you on April 20th. Once again, you are invited to that event and network with many of us that you see here will be at this event. So if you want to see us in person, this is a great place to network and to get ideas, right? And improve it, try to improve yourself. And you don't know who you're going to meet at these events. So I hope you could attend.
Hello.
Have a wonderful day. And thank you for attending our event.
Thank you again.
Thank you so much. Thank you for the wonderful presentation, Isha. And that's it for today. So thanks to both of our speakers and all student presentations. They all give wonderful insight into different streams. I'm very happy and thrilled for the next week also. And our special thanks to Dr. Maria Agapito, our president, New Jersey Academy of Science, and Ms. Donna, Director of Junior Academy, for organizing this wonderful event. Next week, we have a lot of presentation from students from elementary school to high school. Please do join and support our young scientists once again. Thank you. Thank you so much for this, your patience and support. Thank you all.